For this lecture, we will cover the genetic principles in blood banking. So prior to the 20th century, physicians did experiment some with transfusions, and they did this by experimenting with animal-to-animal transfusions as well as animal-to-human and eventually human-to-human transfusions. And it did prove to be very successful. So physicians did kind of experimenting a little bit more with various transfusions. So in the 20th century, there was a great development in the transfusion world. In 1901, Carl Landsteiner did discover the ABO system, and then this kind of led to the advances that started within the world of immunohematology. In the 1940s was when it was kind of discovered the process of adding anticoagulant to blood once it was removed from the body in order to help preserve it and store it for long-term use. Now, all of these advances just over the years of the 20th century did lead to the discovery of other blood group antigens and understanding a little bit more about compatibility and cross-matching and things like that. Now, with understanding the various blood group systems, scientists and physicians did learn that the blood group systems were groups of antigens that were on the red blood cell membrane that shared various related serological properties and genetic patterns of inheritance. So this genetic material is actually found inside of the DNA. Now, the DNA itself is contained in chromosomes that are within the nucleus of every cell that is within our body. Now, the genetic material is then replicated either by a process of mitosis or meiosis, in which that replication produces various genetic material. Now, this all leads to specific patterns of inheritance from the parent to the offspring, which will then demonstrate various phenotypes and genotypes within that individual. Now, what is a phenotype versus a genotype? Well, a phenotype is what the physical, observable expression of those traits are. So in the blood bank world, the patient's phenotype is going to be determined by the particular hemagglutination of the red blood cell antigens when we look at how it responds to a known antisera. So for example, if a person shows no agglutination with either anti-A or anti-B antisera, then we know that there are no ABO red blood cell antigens present or AB antigens present on that patient's cells. So therefore we know that that patient's blood type is type O. So that would be their phenotype, what they present as. With a patient's genotype, now this is their actual genetic makeup. So we can actually determine this by molecular techniques or doing various studies throughout the family to figure out those inheritance patterns. So for example, a patient can have a phenotype of A, but genetically they could have an AA phenotype or an AO phenotype. So in order to be able to kind of narrow that down, we would need to do family studies in order to confirm which one of those genotypes is actually present in that patient. So let's look at the example provided. So let's just say that we have two parents. One parent's blood type is an A and the other parent is a B. So our possibilities for each of these parents would be, of course, the parent could be AA or they could be AO, as demonstrated here with this genotype, or the parent could be BB or expressed as BO. But for this particular example, we're just going to assume that these particular parents have a dominant and a recessive gene, so the dominant of the A and the dominant of the B. So we want to look at the potential outcomes for these two parents for their offspring and what the offspring's genotype could be. So if we know that individuals will inherit a trait from one trait from each parent out of the two possible traits that they can inherit. So in this example, if the mom or father or whatever passed on the gene for A, and then this parent, so you can see here what kind of comes down, and then the other parent passes on the B. So it just kind of goes through all of the various possibilities of what each parent could pass to their offspring and what the potential blood types or phenotype of that offspring would be. So we can see with the probability of two parents having the offspring of between a parent that is a type A and a type B, provided if their genotype has a dominant and recessive gene, that these are the possibilities of the offspring here. Another example here provided is if the parent is type AB and if the parent is type O, then we can see the possibilities. Now, it is a little confusing here as far as the way this is kind of drawn out. So one of the things that have been kind of developed over the years was the use of a Punnett square. So here we have several examples of a Punnett square. And what a Punnett square does is it's used to predict the probability of an offspring's genotype. So if we know what the parent's phenotype is, we can kind of start to narrow down the possibilities of those offsprings. And so the Punnett square will actually summarize every possible combination of maternal and paternal alleles of a particular gene. So it helps us to determine probability of a particular blood type. So in this top example here, we have a parent that phenotype is an AB, and then we have a parent whose phenotype is O. So AB, we know that both of those traits are a little bit more dominant. So we have our A gene and our B gene. And then with our other parents, we have who typed as an O, we know that they have those two O recessives, or they don't have those antigens present for the A and B. So we know that both of these parents are OO and AB. Now, so if this is our AB parent, then we know that they have the gene for an A and a gene for a B, and here's our O parent, gene for an O, gene for an O. So if you kind of come out with these combinations and say, okay, where do these two meet? This is what we get. This particular offspring would be AO, same as this second one. AO, and then this one would be BO, BO. Now, how would they phenotype as far as if we were doing their blood type? Remember that this O is going to be recessive, so that dominant gene of the A would give these particular children or offspring type B. So our possibility for this particular scenario would be 50% of the children would be type A and 50% would be type B. So that's our probability there of that particular offspring. Now let's look at some other examples. This one right here is pretty good, this AB. So if both parents are type AB, what are their probabilities? So we know that we have, looks like 25% would be this type, so type A. 25% would be type B, and then you got two AB, so we know that 50% would be AB. So that's kind of how you figure out your probability based on these Punnett square. So a gene is a basic unit of inheritance on a particular chromosome. So if we look at a chromosome and we look at a specific spot in that chromosome where we're trying to locate more specifically the ABO gene locus. So we look at the particular genetic loci on that chromosome. So here in this particular example, this is where that ABO gene loci is located. So a locus is the site at which the gene is located on a chromosome. Now at those particular locations, you can have alleles that are basically a variation of that particular gene. So what we have at each of those loci is that the antigens are produced by specific alleles that are antithetical. So when we talk about it being antithetical, they are opposite. So you're going to inherit one or the other. Multiple alleles at a single loci are considered polymorphic. This just means that they can have two or more alleles at that particular given location. Now, an example of this that we will talk about will be the Rh gene, because there are so many variations within that particular family or that particular blood group system that this is one that's considered polymorphic because it does have a lot of alleles at that particular location on that genetic makeup. Another one that we can kind of consider polymorphic would be the encoding for the HLA antigens, because there's so many of them, and this is one of those things that they look at when they're doing things like transplants, bone marrows, things like that, because of those matching those specific HLAs because they are so polymorphic. Now, in most cases, when we inherit our blood group antigens, they are inherited in an autosomal codominant pattern, or they have equal expression of both inherited alleles that are found on those autosomes. So in other words, the product of each allele is expressed when you inherit it as a codominant trait. Now, we do have situations in which we can see recessive types of traits. And so this is when a gene is expressed only when inherited by both parents. So an example of this would be the O. When a patient inherits an O from both parents, that O is actually recessive, but it's not going to be demonstrated unless it is inherited from both parents. Now, in our blood type genetics, a recessive trait can actually be expressed as a null phenotype, and so we'll talk about the null phenotypes a little bit later. But this does have to do with the expression of a silent or an amorphic type of gene. So again, that null phenotype just means that when you have certain recessive alleles that are inherited, that they will not express the expected red cell antigen that they are actually coding for. Now, if we talk about a gene being dominant, then that just means that that gene is going to be expressed over the other gene. So this is where we get that AO expresses as a type A. Mendelian principles came after a scientist named Mendel observed certain hereditary patterns in his early genetic experiments that were applied to the study of the blood group systems and the genetics of those blood group systems. So what these principles state is that with independent segregation, the transmission of a trait is in a predictable fashion from one generation to the next. So you kind of know that one gene from one parent is passed to the offspring. This means that your genes will actually segregate, and then it will allow one particular gene from each parent to be passed to each child that are the offsprings of those parents. So another law that must be considered is independent assortment. Now, this is actually demonstrated by the fact that the blood group antigens, that they're actually inherited on different chromosomes, which means that they are expressed separately and in different ways. And what this does is actually gives you a mix of genetic material based off of the different inheritance. So, for example, chromosome 1 is your ABO blood group systems, whereas your KALE blood group systems, they're located on chromosome 7, and so they're inherited independent of each other. So they're still in that blood group antigens, they're just inherited and independent of each other. So you're not going to get the same expression from every parent. And as far as what will be passed on to each individual offspring, so you can have a different makeup of those expressions from child to child. Now, the exceptions to this law are linkage and crossover, which we will discuss a little later. Now, most of your blood group systems are associated, or the genes that are associated with those blood group systems are autosomal. And so what that means is just that the inheritance patterns are the same regardless of the gender of that particular person. Now, the exception to this would be the XG system. So the XG system is actually found on the X chromosome. So what this means is if the father actually carries the XG allele, he will pass that to all of his daughters, but not his son, because as you know, that X chromosome is not passed to the son, just the daughters. Now, if the mother carries that XG allele and not the father, then all of the children will actually express that, because all children get that X chromosome from the mother. Heterozygosity and homozygosity. So if a person inherits different alleles, it is called hetero. Hetero, the root word here, that just means different. So they're different, opposites. So that means that they inherit two different alleles. So you can see in the examples here where this particular patient inherited the A and the O, this one the A and the B, the M and an N, which means that they have both traits for M and N. Now, a person who inherits identical alleles is called homo. So homo root word means the same. So these examples here, you have the AA inherited, the BB, so you have an A from a mother, an A from the father, a B from the mother, a B from the father. With M and M, so you have 2 doses of M from the mother and from the father. So if you would actually phenotype that, where these two are inherited on the same loci, the M and the N, you got the M from both parents, but not the N from either parent. So that is a negative expression, or it didn't happen. So in some blood group systems, persons are homozygous for an allele that have a double dose of that allele. So you can see here they have a double dose of that A, a double dose of that B, and a double dose of that M. So those antigens on those red blood cells are expressed with that double dose compared to those who are heterozygous for an allele. So if they're heterozygous, they don't have as much of that expression of that antigen. What this does is it actually presents a pattern of dosage. So dosage is a variation in that antigen expression due to the number of those alleles present. Now we're going to talk and learn a little bit more about dosage as we go, but first we're just kind of understanding the expression of those particular gene patterns. And what that means is that we will actually have homozygous expression of some of those antigens that will actually show a stronger agglutination reaction when we compare it with antigens from patients who are at a heterozygous type of state or inheritance. So we've got several examples here of patterns of inheritance. So your antigrams, which we discussed in previous lectures, when you see those, it will actually show you, it gives you kind of an idea of which ones are kind of inherited together. So our M, N, and S system, and we will talk about each of these particular blood group systems individually in later lectures. But for now, just kind of notice the patterns where you've kind of got these inheriting get together. So you got your M and N's, your S's, you have Lewis's, your Lutherans, Kales, Duffys, Kids, and these are just some of your more common ones. And of course you have your RH family here. We will talk about that in more detail later. But you can kind of see these patterns of inheritance where you have these heterozygous patterns of inheritance, and then here you have the homozygous. Both parents have given 2 M alleles to this particular patient. Same here with the S. Both parents have given the S expression, not the big S, not the little s. Here you've got the Lewis B, so this patient or donor has 2 alleles of the B, and this one has 2 of the A. Because remember, we talked about these are all specific individual donors. So now you can kind of see the explanation of the heterozygous versus homozygous. So we can expect with our particular antibodies and antigens that have the different expressions. If a patient is presented with an antibody and we get reactions, those expressions can be stronger on the particular reactions where you have those homozygous type of expressions of those antigens. Now sometimes you can have genes that can interact with each other, and of course it will depend on whether or not they are inherited on the same chromosome, so this is a cis expression, or on an opposite chromosome, so this is a trans expression. Now, these interactions may weaken the expression of one of the antigens that's actually encoded by the particular genes. So, for example, the CE and the G genes of the Rh blood group system are actually inherited on different genetic loci. So the CE gene encodes the C, or the big C, and the little e antigens. And then the D gene actually encodes for the D antigen. Now, when the CE is inherited in trans to D, it actually weakens the D antigen expression on that particular red cell. Now, in some of your blood group systems, these antigens are actually encoded by 2 or more genes that are close together on the same chromosome, and they are inherited from each parent as an actual whole unit. So genes that are so close together on a chromosome that they are inherited as a unit are considered linked. So independent assortment does not occur when these genes are linked. These gene units are called haplotypes. Now, for example, within the MNS blood group system, the M and the N are alleles on one particular gene, and then the big S, little s are alleles that are located on another gene. But because these genes are so close, they are actually inherited as a haplotype. Now, these haplotypes, they actually happen within the population at a different frequency than would be expected if the genes were not actually linked together. Now, linkage disequilibrium refers to the phenomena of antigens occurring at a different frequency within the population, and it's actually depending upon whether they were inherited by linked or unlinked genes. So in other words, if the M and S genes were not linked, the expected frequency of those M and S antigens in the population would be 17%. But because of this particular linkage disequilibrium, they are actually observed in a frequency of the MS haplotype is actually more closer to like 24%. Now, the other exception to that law of independent assortment is that you have something called crossover. And so what happens is you have 2 genes on the same chromosome that will actually recombined. And they do this in this crossover state in which they exchange this genetic material during the meiosis after the chromosome pairs have actually replicated. And so what this results in is that those genes are exchanged during this process, but they're not actually lost. So the recombination results in 2 new different chromosomes. Now, if we would want to determine a particular genotype or phenotype occurrence in a particular person as far as what that occurrence would be for that particular genetic makeup, then we've got a couple of formulas that we can actually use to determine that. And we did go over this a little bit within our laboratory math of how to do these calculations, but we're going to kind of review that again. So a phenotype calculation actually enables you to be able to find a particular unit of red blood cells based off a certain antigen characteristic. So it kind of lets you know how many units you would need to screen to get the particular unit you would want. Now, the Hardy-Weinberg formula calculates a determination of the gene frequency that is produced by a particular trait. Phenotype calculations are done by knowing the frequency of an antigen expression. Now, we'll talk about each individual antigen and their expressions and things like that a little bit later, but for now, we're just kind of talking about the calculations to get there. So, for example, this particular patient was tested and found to have 3 different blood group antibodies. And so those antibodies are to the big C antigen, the big E antigen, and the big S antigen. Well, this patient needs blood, so we know if these antigens are present on the red cells, then we've got to find units that are negative for those particular antigens, because if the patient has the antibody, then we know that that antibody is actually going to agglutinate that particular antigen. So we need a red cell that is negative for all of those particular antigens. So what we do is we kind of calculate based off of frequency. So what is known? We know that out of the population of donors or individuals or people, that this is pretty much the percentage of the population that will present with those particular antigens. So we know that 70% will present with a C antigen, 30% with an E antigen, and 55% with an S antigen. Now, so if we know that these are this group of patients that have those antigens, we are looking for this group. We want to know the ones that do not have that expression. So out of our population, if 70% have it, then we know that means that 30% should not have it. 70% should not have E, and 45% should not have S. So this is the percentage of the population that are negative for all of these antigens. Now, are they negative for all three? Well, we don't know. But we know by doing some calculations that we can figure out what percentage, based off of this percentage of being negative, how many of those patients would actually be negative for all three. So what you would do is you would take those percentages of those populations and you would multiply all of them. So doing that multiplication would give you this here, which is less than, if you do your rounding to significant figures, that would give you about 10% of the population would be negative for all three. So what does that mean if 10% are negative for all three? That means that if you did test at 100 units, then that means that 10% of those would be negative. Now, if you kind of reduce that math, that means 1 in 10 units would be actually negative and compatible for that patient. Now, let's take that a step further. So let's just say we have another patient, same scenario, but this time we know that the physician has ordered 2 units of red blood cells to be transfused to that patient. So here we know that the patient has antibodies to Duffy A, JKB, or your KID B, and your KALE. So if we know the frequency of how many patients present with those antigens on those red cells, then we also can figure out the percentage of donors that will present with that negative makeup. So we take all of those percentages and then we multiply them out, so you can see where that was done here. And then once you multiply it out, you get about 9% of your units would be negative, or 9 out of 100 units. But we know that 2 units are needed. So what do? We take that 2 that we need and we divide it by that percentage of negative. And that gives us a frequency that we would need to screen 22 units in order to find 2 units that would be compatible. Now, an English mathematician named Hardy and a German physician named Weinberg, they kind of come together to develop a concept of genetic equilibrium. Now, they did this independently, but what happened is their theories led to the formulation of the Hardy-Weinberg law. And so what this is a statistic formula that was derived from their principles that estimates the frequency of genetic diseases and observable traits. So this formula is based on the principle that the sum of the gene frequencies when expressed as a decimal is equal to 1.0. So what this formula actually does is it's used for predictions of populations at equilibrium, meaning that there is no migrations or mutations or natural selections that's happening. And it's also based off of random mating, so just random people producing offspring. So there's not any kind of directed reproduction. So by observing the phenotypes or the traits in a large number of individuals, the percentage of the traits actually occurring is then established. So what this formula does is it actually calculates a determination of gene frequency that is able to produce a particular trait. So the probability of heterozygous and homozygous expressions for each of the genes in a particular system can be actually predicted. Now, with new advancements and the ability to actually characterize genes and determine the zygosity of a particular individual, we can do this through molecular methods now. So these formulas aren't really used as much, but they still can be applied into the world of blood bank as far as trying to determine the frequency of inheritance for homozygous and heterozygous. So here we have an example. If we want to know if we're looking at a particular allele to inherit it, big A, and of course we represent that by P. And then if we want to know little a as far as the frequency of it on a particular allele, then we will look at P. So what is the frequency of Q if P is 0.3%, so, or 0.3 or 30%? So if we know how that is inherited, then we can determine what would be the frequency of Q. So this is our unknown. So if we plug everything into the equation, so 1 minus this particular frequency, and that would give us 0.07. So what are the genotype proportions? So we know that AA, or to inherit the homozygous for A, if we did P squared, that would give us a 0.09. If we want to know the heterozygous inheritance, then of course we would work that out to give us 0.42. And if we want to know the homozygous for little a, then we could work that out and it would give us the 0.49. So this is how we derive those frequencies of inheriting in a homozygous versus a heterozygous for each of those. Now, as I previously mentioned, the fact that there are different molecular genetic applications now that kind of can help with that. Some of these things aren't as frequently used. But depending on where you're at will depend on whether or not they actually do any of this molecular genetic testing. You're not seeing it so much in some of your just hospital blood banks anymore, or in your hospital blood banks. You're going to have to get to some of your bigger facilities where they're actually doing transplants and things like that before you're going to see some of this testing. So I'm not really expecting you to know this. Just be aware that there are newer methods that are being developed to try to get matches that are more closely matched based off of that molecular patterns of inheritance and things like that. So getting down to that deeper DNA molecular level to actually get a better match for that particular patient. And that concludes this lecture.